Another aspect of immune cells is the presence of pattern recognition receptors. Pattern recognition receptors are little sensors within the cell that detect signs of invading microbials or viruses and therefore influence the body's specific immune response. They do this by detecting microbe associated molecular patterns, pathogen associated patterns, and damage associated molecular patterns within the host cell itself. And they do this by monitoring the material ingested by the cell, outside the cell, and within the cell's cytoplasm. So pattern recognition receptors that monitor the material ingested by the cell are called toll-like receptors. They're located in the cell's endosomes or phagosomes, and they face inwards towards the inside of the organelle. So they're able to monitor and detect microbial nucleic acid within the cell. There's two types of pattern recognition receptors that monitor a cell's surrounding. The first one is also called toll-like receptors. They're found within the outer membranes of the immune cells, in their endosomes and in their phagosomes. They detect the presence of pathogens by characteristics of their outer surface, such as liposaccharides, lipoprotein, and flagellin. And then detection of those triggers an immune response within the cell. The other type is C-type lectin receptors. These are located purely in the outer membrane of the immune cells, and they detect pathogens and bind to carbohydrates on their surface. And by doing this, they're immediately able to inform lymphocytes, which shapes the cell's immune response. And then finally, there's pattern recognition receptors within the cell's cytoplasm. There's two types. There's RIG-like receptors, which detect viral RNA and notifies other cells about the presence of a virus. Or there's NOD-like receptors, which detect components of invading cells and damage within the host cell itself. This specifically triggers an inflammatory response within macrophages and dendritic cells. And these pattern recognition receptors are very important because when they do detect viral cells, they trigger the release of an infron to other cells. And this causes those outside cells to produce antivirals. And when these antivirals are activated by the presence of an invading virus, it signals for the host cell to degrade their mRNA and stop protein synthesis, which just leads to the cell's death. And so, therefore, the virus isn't able to spread anymore because it was never fully able to inhibit the cell itself.
When a host cell recognizes that it is being infected by an invading microbe, the cell will produce infrons, which is a type of protein that will warn nearby cells about the invading microbe or disease. In response to the sensor signals that indicate tissue damage or bacterial infection, phagocytes will go to the site of damage or invasion. Phagocytes are cells that are specialized in engulfing and digesting cell debris and microbes. This is a process called phagocytosis. Some types of macrophages also play roles as sentinel cells, which is the body's first line of defense. If multiple sensor systems find infection or tissue damage, then a set of events called inflammatory response will occur. With this response, changes to local blood vessels allow fluids to leak out of the tissues, with those fluids containing complement system components and other proteins. These proteins and other signals attract phagocytic cells, which move from the bloodstream to tissues. Another response is fever, which is a rise in temperature that's higher than normal. There are many cells involved in phagocytosis and inflammation, which would include in neutrophils. These phagocytes are essentially microbe-destroying machines. Their granules contain many destructive enzymes and antimicrobial substances. Neutrophils can also degranulate to kill nearby microbes, bursting in the area of infection. In eosinophils, they rid the body of parasitic worms and are involved in allergic reactions. They phagocyte antigen-antibody complexes and also have granules that contain multiple antimicrobial substances that can be released by degranulation. Basophils are involved in allergic reactions and inflammation. Their granules contain histamine and other chemicals that are involved with inflammation. Monocytes develop into macrophages, which means large eater, showing their role as a type of phagocyte. They kill microorganisms, stimulate the action of other immune cells, and remove dead cells. Dendritic cells engulf material in the tissues and bring them to the adaptive immune system for inspection. There are three lines of defense against invading microbes, inflammation being the second. Inflammation is caused by cytokines used as the voices of a cell. Cytokines act at extremely low concentrations, which have local, regional, or systemic effects. Cytokines made by one cell diffuses to another and will bind to its appropriate receptor of said cell. The binding induces a charge in the cell, such as growth, differentiation, movement, or cell death. For inflammation, this includes interferons. They have an antiviral effect and are important in regulatory mechanisms, stimulating the responses of some cells while inhibiting orders, like NK cells. NK cells will just go and basically tell a cell to destroy itself. Interleukins and macrophages in epithelial cells. They promote inflammation, induces fever, activates macrophages, and causes T cell activation. In T cells and macrophages, they contribute to inflammation, induce fever, and have T and B cell proliferation. Tumor necrosis factor, or TNF. They, in macrophages, T cells, and NK cells, they promote inflammation and regulate certain immune functions. They play a role in killing tumor cells. Overall, inflammation limits the spread of pathogens and destroys them, initiates tissue repair, and removes debris.
I'm going to be talking about the complement system and fever. The complement system is a series of proteins that routinely circulate through the blood. These proteins circulate in their inactive form, and then they will be activated once they sense the presence of a pathogen, which will start a chain of reactions between the proteins involved in order to destroy that pathogen. There are 9 proteins involved in the complement system. They are labeled numerically. They all just start with C for complement, so it's pretty easy to figure out which ones are which. It's just C1, C2, C3. And these proteins can also split into 2, so in that case it would be like C3A or C3B. These proteins are not listed in any order specific to how they react. It's just how they were discovered. And then there are 3 different pathways that activate these complement systems, and that's alternative, lectin, and classical pathways. So, like I said, there are 3 different complement system pathways, and all these pathways will result in the formation of C3 convertase, which then can differentiate into different splits of that C3 convertase that go into different reactions that help kill pathogens. That is like an inflammatory response, opsonization, and the lysis of foreign cells. So first pathway is the alternative pathway, and this is not because it's the second choice, it's or anything like that. It was just discovered after the classical pathway, and that's why it's called the alternative. The alternative pathway is triggered by C3b binding to microbial invaders. And then we have the lectin pathway, just triggered by Manrose binding lectin binding to microbial invaders. And then there's a classical pathway, which is triggered by antibodies binding to microbial invaders. And like I said, all these go into the formation of C3 convertase. And then there's 3 major outcomes, which I said before, that occur after the formation of C3 convertase. The first one is opsonization, which is where C3 splits into C3b and prepares bacteria basically for eating, you could say, or think of it like seasoning it so that the phagocytes want to eat it more. Basically, they're just being added with opsonin is getting added to the bacteria so that the phagocytes are more likely to get rid of them. And then there's the inflammatory response, which is C3 splitting into C3A and C5A, which attract phagocytes to the area and trigger mast cells to release histamine, which causes inflammation and just causes more phagocytes to come and see what's going on and clean up the bacteria. And then there's also the lysis of foreign cells, which is C5b combining with protein C6, C7, C8, and C9, and they form a membrane attack complex that inserts itself into a bacteria's membrane and destroys it, basically. And then I'm going to talk about fever responses. A fever is a part of our inflammatory response that causes an increase in our overall body temperature. Bacterial infections often lead to fever, which is seen as a bad thing, but this actually isn't a bad thing because it means our body is working to fight the fever. If we didn't have a response, that means our body would not be reacting to a bacteria in our body at all, which would be really bad because then the bacteria would continue to grow and live. And then fever in the body signals leukocytes that there are pathogens harming the body that they need to stop.
There are 5 types of antibodies. Antibodies are glycoproteins, termed as immunoglobulins, and they are produced in response to an immune reaction and specifically bind to antigens responsible for initiating that reaction. The 5 antibodies are IgG, IgM, IgA, IgE, and IgD. IgG accounts for 70 to 75% of all human immunoglobulins found in the blood. IgM is the largest antibody and the first one to be synthesized in response to an antigen or microbe, and it also accounts for 5% of all immunoglobulins present in the blood. IgA accounts for 10 to 15% of immunoglobulins and is prevalent in nasal mucus, saliva, breast milk, intestinal fluid, and serum. IgE is the least prevalent one, with a serum concentration 10,000 times lower than IgG. However, the concentration of IgE increases significantly in allergic conditions. IgD functions as a B cell antigen receptor and may participate in B cell maturation, maintenance, and so on. There are several outcomes of antigen-antibody binding. One of these outcomes being opsonization, which is a key process in the immune system by which pathogens or other foreign substances are targeted for destruction by phagocytic cells. This process involves the coating of these pathogens or foreign substances with opsonins, such as antibodies or complement proteins, which serve as ligands for phagocytic cells. Another outcome of antigen-antibody binding is complement system activation. What happens here is immune complexes trigger the classical pathway, and specific proteins of the complement system attach to the Fc region of the attached antibodies. Another outcome is prevent adherence and immobilize, which is where antibodies can block or immobilize microbes to prevent infecting host cells. Neutralization also occurs in antigen-antibody binding, which is where the toxin/virus is coated with antibodies, preventing attachment to other cells. And lastly, agglutination is another outcome, which is where antibodies can bind to multiple antigens, cross-linking them for phagocytosis in large groups.
What's up, you guys? My name is Emmanuel, and today we're going to be talking about my CLC presentation in terms of the Christian review part. So, the verse that I picked today was from Ezekiel 34:16, and it says that I will search for the lost and bring back the strays. I will bind up the injured and strengthen the weak, but the weak and the strong I will destroy. I will shepherd the flock with justice. So basically, like, I feel like what this verse is trying to say is that, like, God will always have, like, some a way for us to, like, help, like, the weak and, like, those who are injured. So basically, like, taking into the fact of, like, as, like, a nursing student, I feel like God like gave, like, certain people the responsibility to be, like, nurses. And I feel like he gave those responsibilities to those people that, like, have the passionate and, like, determination to, like, care for those people that who are in need, like, especially who are sick, like, especially who are injured. And I feel like with those type of, like, people in this profession, I feel like it's really important. It's because, like, taking care of, like, someone else's, like, health is a really huge responsibility because their life is depending on it, and which could lead to, like, so many factors in their life, and just, like, having to like take care of their their health, and taking into the account as well into the microbiology side. I feel like God like also like gave certain people like responsibilities for to study microbiology as like a way to like understand like how like certain like bacterias or like pathogens like affect our body, which helps us to like figure out and study and analyze like things that like a treatment for that in order to like improve our health like in the long run as well. And I feel like the importance of this is that it actually helps us to like improve like stopping like bacteria from spreading, and I feel like that's like what I found like the most important thing about with this verse.